One here is cellular energetics, topics 3.4 through 3.7. Photosynthesis, cellular respiration, get into cellular energy and how it's used. I'll keep this up here so you can see the topics and post this Word document to the video link as well. And there'll be some questions throughout. All living systems require a constant input of energy. You should be familiar with the ATP-ADP cycle. These two diagrams here are the same. Different levels of detail. Remember that ATP is the triphosphate shown here with 1, 2, 3 phosphates. ADP is the diphosphate. These phosphate to phosphate bonds are high energy, so the more high energy bonds there are, the greater amount of energy that's going to be stored here. You should recognize ATP and ADP as what macromolecule? That is correct. A nucleotide. It is an adenine base, the same adenine base that's in DNA or RNA. It's ribose. Ribose is the pentose sugar that's found in RNA, and then it has 2 phosphate groups or 3 depending on its energy level. And it's constantly getting put back and forth. When energy is input in here, in other words, when a phosphate is added, a bond is made. Making bonds is going to capture energy or absorb energy. Making bonds absorbs energy. So adding a phosphate group, absorbing energy, that energy then is stored to do work later. Breaking a bond, such as using ATP and reducing it down to ADP, breaking a bond is going to release energy. And so many times this happens where one molecule is broken and that powers the reaction for another molecule. And that's called coupling. What are some sources of energy? Well, we think the sun certainly is a major source of energy, but sugars are sources of energy, fats as well, macromolecules. Proteins can be in extreme cases, but for the most part, it's going to be sugars and fats along with ATP molecules. Metabolism is, of course, all of those reactions. Breaking and making bonds in your body is metabolism. Anabolic is the building up. Catabolic is the breaking down. Both of those together make metabolism. Energy input must exceed energy loss. What does that mean? Well, every time energy is transferred from one form to another, so when solar energy becomes sugar or when sugar becomes ATP, not all of the energy is completely transferred. It's not 100% efficient. Most of the time, it's barely efficient. It's 10, 20, maybe 30% efficient at best, which is that energy is going to be lost as heat. Part of the thermodynamics laws. Every time energy is transferred from one source to another, much of it is going to be lost as heat. And so when you have a reaction that releases energy, that reaction then can then be used to fuel other reactions that may require energy. Or you may have this cascade effect where one reaction powers the next reaction and then that powers the next reaction. And so a lot of times, and especially in these things, the product of the first reaction will become the reactant of the second reaction, and then the product of the second reaction becomes the reactant of the third, and so forth and so on. So you should be familiar with labeling of a reaction. You've got two sides here, the reactant side and the product side, and the arrow points to the product side. That's a little bit of review that hopefully you didn't need, but okay. Identifying reactants and products, the arrow points to the products. Of course, you've got to have the same number on each side. There's a conservation of matter, not really a major part of AP Bio, but it does help to understand that you're going to have, if you have one carbon on this side, you'll also have one carbon on that side. Same number of oxygens, hydrogens, etc. Where do we see ATP or energy coupling a lot? Well, ATP, when it's broken down, high energy, breaking a bond here means that we can make a bond here and turn these reactants into other products. And now we've reduced ATP down to ADP, and it's going to go back and get recharged. This happens in photosynthesis. This happens in cellular respiration. It happens, in any reactions that require energy, like active transport, movement, any of those other ones. All right, photosynthesis. The highly complex organization of systems. Of living systems require constant input of energy and the exchange of macromolecules. So we'll look for energy here, and we'll look for what macromolecules are being passed. Describe the photosynthetic process that allows organisms to capture and store energy. Photosynthesis captures energy from the sun and produces carbohydrates. So here we are taking reactants that really don't have a lot of energy, and we are upgrading them. In other words, we're making more bonds. So more bonds are going to be made than are going to be broken. That's why this reaction is going to be a net gain of energy or a net absorption of energy, because we're going to turn solar energy, the energy of light, into chemical energy, the energy of electrons in bonds, right? Chemical energy is about bonds, the shared electrons in a bond. And we've made a bunch of bonds here in glucose compared to carbon dioxide and water alone. Photosynthesis first evolved in prokaryotes. Remember, prokaryotes lack membrane-bound organelles. They don't have a chloroplast, so they must have done photosynthesis without a chloroplast. And part of the understanding of chloroplasts is that they show evidence of being previously prokaryotic organisms because of their type of DNA and ribosomes. It is believed that photosynthetic prokaryotes like cyanobacteria are responsible for creating oxygen in the atmosphere. Early Earth's atmosphere did not have oxygen, and you can see that with oxidation of rust in rock formations. Today's atmosphere is about 21% oxygen gas, so we went from 0 to 21%, and that first was prokaryotic photosynthesis. So cyanobacteria are a group of bacteria that's still around today that do that. Prokaryotic pathways led to the foundation of eukaryotic pathways, and here's a shot of the chloroplast with its thylakoids, these interconnected stacks. Each stack is called a granum. A grana is plural. Light comes in. These are green because they contain chlorophyll. Chlorophyll is the primary photosynthetic pigment. And if I blew this up, white light here, the light from the sun, is really a combination of the visible light spectrum, which is the Roy G. Biv, and what is reflected here is the green. What is absorbed are the two ends of that spectrum. The reds and the blues are absorbed, and the green is reflected. Photosynthesis is broken down into two stages. The first stage is called the light-dependent reaction. It can also be called the light reaction. The second stage is called the light-independent reaction. Could also be called the Calvin cycle. I remember when I was in high school, we learned it as the dark reaction, but it does not take place in the dark. It just does not use light directly. It uses the energy captured from light, but at this point it's chemical. So what do we got going on here? The light reaction here on the left is about absorbing light energy, that's going to be with chlorophyll, and upgrading these intermediates or these electron carriers. So we have ADP and NADP. These are, NADP is a coenzyme. It's a molecule that's going to be carrying electrons. And of course, ATP or ADP is a nucleotide. And it's going to be the energy currency of the cell. So what does the light reaction do? It captures light, turns it into chemical energy, and in the process will split water and produce oxygen. So where does oxygen come from in photosynthesis? It doesn't come from CO2. It comes from water being split. Now, it's important to remember that when the water is split, there's not just oxygen left over, there's hydrogen ions and electrons, and those are going to fit into this cycle here. And ultimately, they're going to get passed into sugar. The light-independent reaction, or the Calvin cycle, is about taking CO2 out of the atmosphere, which is inorganic, and building up an organic molecule like a sugar. And how does it do that? Because this is a reaction that requires a lot of energy. Well, it's going to use the energy from the light reactions. And so now ATP and NADPH are going to be downgraded back to their low energy forms. And so if we scroll back to the ATP-ADP cycle, well, that's really an example of an ATP-ADP cycle where energy is being upgraded and downgraded based on its use. During photosynthesis, chlorophyll, and there's other pigments as well. Think back to the paper chromatography lab that we did where we separated chlorophyll from other pigments, and we saw that there were pale yellows and even like a little bit of a orangish color, and then there were the greens. So chlorophyll is the primary pigment, but not the only pigment. Absorb light, and then they're going to boost electrons to higher energy levels. So here is a diagram that is inset in, so this diagram here, let's see if I can get this diagram here would be inside the membrane of the thylakoid. And what do we got going on? Well, we have these things called photosystems. The diagram is complicated, but I think what you need to understand is the basic connection between these. Photosystems are a series of proteins in the membrane where that chlorophyll is found, and electrons are going to be passed. Okay, so the reason this is photosystem 2 followed by photosystem 1 is this one was the first one to be discovered, and they never changed the name. So light comes in, excites an electron, so a photon of light will hit this molecule. Chlorophyll is a very big molecule. It gets hit and it loses an electron, sends it up to a higher energy level. Well, if electrons were continuously being lost, then chlorophyll would eventually run out of electrons. But what does it do? It replaces electrons as they get passed. And where do they come from? They come from water. Remember, water is split. If we split water, we end up with electrons, hydrogen ions, and oxygen. Oxygen is released. It's a product. Electrons are passed down a series of proteins, and then the hydrogen ions are going to be used to create a proton gradient. In photosystems 2 and 1, these are embedded in the membrane of the thylakoids. They're connected by this a series of proteins in the membrane. Now you can't see the membrane here, the phospholipid bilayer, but it's there. And they're going to be connected by this structure called a electron transport chain. And don't get confused or worried about the proteins, the names cytochrome or PC and PQ. Electron transport chain is a series of proteins always in a membrane that pass electrons from high energy to low energy, and it works kind of like a water wheel in a river. So the water wheel in the river turns as water flows. This is going to create an electron gradient based on, proton gradient based on the flow of electrons. And ultimately ATP is going to be produced. So where do we see the things that come out? We saw what went in, water went in, and light went in, but what comes out? Well, ATP comes out, and hydrogen and these electrons come out. So here we end up with NADPH and ATP. And if we scroll back up to the diagram here, what do we see come out of this? ATP, NADPH, and oxygen. And what did we see come in? Light and water and NADP and ATP. Here's a more realistic, I guess, picture or diagram of this. So this is those green thylakoids. This is the space in between. The space in between is called the lumen. And this is where these photosystems are connected. Hydrogen ions are passed in. Right? These hydrogen ions are passed in. This, as the concentration increases on the inside, the pH is going to drop. Right? More hydrogen ions means more acidic, lower pH number. Light hits the photosystem, excites an electron. Electrons get passed, and notice it's kind of look like it's going downhill, and that's shows you that they're dropping in energy level. And then light hits photosystem 1, excites the electron again, and it gets passed on to this enzyme, NADP reductase. So it's reducing NADP. Remember, anything that gains an electron is reduction, and now we end up with NADP and ATP. So how did ATP start? Well, there's this really important molecule, which we'll talk about in cellular respiration as well, that's called ATP synthase. It's an enzyme, ASE, right? And it adds a phosphate group to ADP. How does it do that? Well, electrons pass down this chain, creating a hydrogen ion concentration gradient, and as they flow down their gradient, high concentration to low concentration, that doesn't require any energy, but it is going to power ATP synthase. So when electrons are transferred between molecules in a sequence of reactions as they pass through the electron transport chain, an electrochemical gradient of protons is established across the inner membrane. So now we have a high concentration here, low concentration here. These will diffuse through ATP synthase, and the type of diffusion that is called chemiosmosis. The thylakoid shown up here in this kind of gray-brown space has more protons, lower pH, than the surrounding stroma, that's the fluid on the outside. The formation of the proton gradient is linked to the synthesis of ATP from ADP with this enzyme. Here's a kind of a simpler drawing without any of the electron transport chain proteins shown. But you can see that if this has a high concentration and outside is low, the hydrogen ions will flow through the gradient, and this powers that enzyme. Chemiosmosis is the flow of protons, hydrogen ions, through the enzyme ATP synthase, which powers the addition of a phosphate group to an ADP molecule. All right, now we've got ATP and NADPH. These are high energy molecules that will go to power stage 2, which is the Calvin cycle. The energy captured in the light reactions and transferred to ATP and NADPH, which are the electron carriers, powers the production of carbohydrates in the Calvin cycle. And there's this diagram again. Inorganic oxygen or carbon in the form of CO2 to organic carbon in the form of sugars. This takes place in the stroma. Here's a blown up version of the Calvin cycle, and you can see that ATP is used and NADPH is used, and there's a series of reactions that go add carbon, carbon, carbon until you end up with the output of glucose. You do not need to know the Calvin cycle in this detail, just put it in there for some context. And just kind of read through some of those. Remember, the memorization of the steps of the Calvin cycle or the structure of these, the names of these enzymes, is beyond the scope of this class and exam. All right, cellular respiration, topic 3.6. Describe the process that allows organisms to use energy stored in biological molecules, such as carbohydrates, lipids, proteins, and nucleic acids. Here we have the mitochondria and cellular respiration. We can see that there are some steps for cellular respiration, starting with glycolysis. This is sometimes referred to as the link reaction, and it connects glycolysis to the citric acid cycle, which is also referred to as the Krebs cycle. And then the last step here is oxidative phosphorylation, electron transport chain, and chemiosmosis. This is the exact same as photosynthesis. All right, fermentation and cellular respiration use the energy from macromolecules to produce ATP. These processes are found in all life forms. So this is some continuity. This is some evidence of common ancestry, the fact that all life forms have fermentative and cellular respiration pathways. Now in eukaryotes, it's going to be more complex because prokaryotes lack a mitochondria. And this is going to involve a series of reactions. They're going to be enzyme catalyzed, which means they're going to be highly controlled. Let's talk a little bit about the electron transport chain again and see if we can see some connections. Where are electron transport chains found? Well, the chloroplasts have them, and that's the all those spaces on the inside, all that thylakoid that increases the surface area, and therefore allows a lot of these to exist. Mitochondria, highly folded internal membrane. If it's highly folded, that means that it can have a lot of electron transport chains and therefore have a high efficiency. And this is also found in the plasma membrane of prokaryotes. They have a way to pass electrons as well. Electrons delivered from carriers, in this case it's NADH and FADH2, they're passed through a series of electron acceptors. Those are proteins found in the membrane. And as the electrons pass, they go towards their final destination, their final electron acceptor, which is oxygen. I'll go ahead and put that in bold. Oxygen is the final electron acceptor for eukaryotic cellular respiration. There are some exceptions to that, like in photosynthesis. NADP plus is the electron acceptor, and then anaerobic bacteria, those that do not use oxygen, are not going to have oxygen. They're going to use other molecules. The electron transport chain creates a proton gradient. We've seen that. Creating a hydrogen ion concentration gradient is like charging a battery. So you have this potential energy. We have high electrons in one, or high hydrogen ions in one area. On the other side of the membrane, low hydrogen ions, and they're going to want to flow downhill. And that is free energy if you can establish the gradient. The flow of these protons down the concentration gradient is called, you guessed it, chemiosmosis, and it is going to power the enzyme ATP synthase. So what are the steps in glycolysis, or in cellular respiration? Number one is glycolysis. This is the pathway that takes glucose, a 6-carbon sugar, and breaks it into 2-pyruvate, which is a smaller sugar made up of 3 carbons. But there are some other products here too. Electron carriers, NADH is produced, and ATP is also produced. Actually, 2 ATPs are produced for every 1 glucose. What is the fate of the NADH? It will go and get oxidized during the final steps here, if oxygen is present. This takes place, and glycolysis takes place in the cytoplasm. It does not require oxygen. Therefore, it's an anaerobic process. It does produce 2 pyruvates, and it does produce 2 ATPs and 2 NADHs. Pyruvate is then transported into the mitochondria where it can be broken down or oxidized further. You can see that here, pyruvate oxidation with acetyl-CoA. This is a coenzyme. It helps these reactions. This is sometimes called the link reaction. But generally, I feel like AP biology just considers 3 steps here, which is glycolysis, citric acid, and oxidative phosphorylation. The citric acid cycle is also called the Krebs cycle. Carbon dioxide is released. Pyruvate is broken down into all these different intermediates. ATP is produced. But more importantly, electrons are going to be transferred to NAD+ and FAD2+, so that they can go on and get oxidized at the end. And then finally, the payoff here is oxidative phosphorylation. The electron transport chain and chemiosmosis take the electrons from NADH and FADH2, and they pass them down the electron transport chain to create a hydrogen ion concentration gradient that will power ATP synthase, and we can see that here. So here is NADH moving up, dropping off its electron, and now we have NAD that can go back and get recharged or reduced. FADH2 turns back into FAD, and it's going to go back and get reduced again. And then the yellow arrows represent the electron pathways that they get passed between these different proteins in the electron transport chain. And now, when this is happening, all those little hydrogens that got dropped off will get pulled across the membrane, establishing a hydrogen ion concentration gradient. And then they will freely diffuse down ATP synthase, which is an enzyme, but it's in the membrane, so it acts like a transport protein. And as hydrogen ion moves through, it will power this enzyme, turning ADP into ATP. All right, here's kind of this connection where you can see all of the numbers. NADH, FADH2, how many of them are produced, how many ATPs are produced per glucose molecule. All right. Fermentation. I'll post this diagram too. Fermentation allows glycolysis to continue. Fermentation will take place if oxygen is absent, so if it is anaerobic. Glycolysis will always occur whether or not oxygen is there or not. And what happens to pyruvate will be determined on if oxygen is present. Now the pathway here, the aerobic pathway with oxygen, is preferred because it is going to produce so much more ATP. If oxygen is absent, no more ATP will be produced after glycolysis, and it only produces 2 ATP. Ethanol and lactic acid are the other products, depending on what organisms do this. For example, animals produce lactic acid. Plants, bacteria do alcoholic fermentation, yeast. All right, let's look at a sample question. Researchers studied the relationship between glucose concentration, oxygen level, and ATP production in a type of mammalian cell. Cells were isolated and cultured in a growth medium containing either 1.5 micromole of glucose or 25 micromoles of glucose, and at oxygen levels that varied from 0 to 21%. Remember, 21% is current atmospheric oxygen, high oxygen level. The researchers determined that the concentration of ATP per cell under different concentration conditions, and we can see that here, how much ATP is made per cell. That's the results there. They're shown here as relative to the maximum ATP concentration obtained when cells were cultured in the presence of 25 micromoles of glucose and 21% oxygen, standard culture conditions. So here we have some data, and you're going to be asked to do a couple things with this data. Number one is describe the role of oxygen in cellular respiration, construct a graph with appropriate titles and labels to represent the data in Table 1. Describe the relationship between concentration of glucose in the culture medium and the ATP concentration in the cells. And then finally, the application here is in a further experiment, researchers add a component to the cell growth medium that both binds and releases protons or hydrogen ions and is allowed to pass through the lipid membrane. So it's kind of an interesting thing there, because normally hydrogen ions cannot pass directly through the membrane because they're charged. They must go through a transport protein. Predict the effect of this, of adding this compound on ATP production by the cells. Justify your prediction. All right, so go ahead and pause the video and answer those 4 questions, and then we'll go over them now. Okay, welcome back. Describe the role of oxygen in cellular respiration. Let me just roll back up here to tell you that oxygen is the final electron acceptor, right? Oxygen is the final electron acceptor. Without oxygen, all of this electron transport chain comes to a halt. You can see here is oxygen, right? Here is oxygen right here, accepting the electrons. And of course, when you add oxygen, hydrogen ions, and electrons, you end up with water, which is why this is one of the products of cellular respiration. So part A, very easy. Construct a graph with appropriate labels and titles. All right, this is very likely what they're going to ask you to do on the test in some capacity. I would imagine that they're going to have you build a graph. So let's practice. Hopefully you got a graph that looks like that, and we'll look over it in a minute. But when you see a data table like this, the left-hand column is going to be your independent variable, usually. And so here you have 2 separate conditions, which are the 2 separate ones, and this is going to be the x-axis, because you kind of have 2 separate independent variables. And here is your graph, the percent oxygen at the different sugar levels. And then your dependent variable is going to be your right-hand column, which is going to be what they measured. Remember, it says up here that the researchers determined the concentration of ATP per cell under different conditions. What is being measured, what is being determined, that is your dependent variable, and that is your y-axis. So you really have, you could have done 2 graphs. I would imagine they would accept 2 graphs. Where you have 2 different ones, but obviously you want to put these on the same graph because they're going to ask you to compare them. So here we have percent oxygen and relative concentration of ATP per cell, and then the 2 different line graphs show the 2 different conditions. And do they look similar? Yes, they do. Notice we have a title, relative concentration of ATP at different growth conditions. You can just take that right from the data table. Got to have a title on there. X-axis here is the independent variable, the percent of oxygen. The y-axis is the relative concentration of ATP. That's your dependent variable. And again, this table really shows 2 separate independent variables, but you can see that's what we got. And that looks good. The key shows which one is which graph is which curve, and you could label them or color them independently. Just so you show the difference. You could even, if you're doing this, put a line that shows which one is which. Describe the relationship between concentration of glucose in the culture medium and the concentration of ATP cells. So these are the two different sugar concentrations. The sugar concentration does not drastically change the relative amount of ATP, at least not. It does by a little bit. It's higher, but not by much. So that would be my conclusion there, is that the amount of glucose, it's so much more glucose, but it really is barely more ATP, if you can see that. And that's kind of the conclusion. In a further experiment, this one's the most complicated part, I feel. Researchers add a compound to the cell growth medium that binds both binds and releases, so it's going to grab hydrogen ions and let them go, and it passes through the membrane. So if we think to our the role of hydrogen ions in this whole process, if a compound binds to protons and passes through the membrane, it's never going to allow a concentration to build up. So here's what it is in normal conditions, where hydrogen ions are high and then they're going to flow through. But imagine we have a compound in here that will grab these and pass directly through the membrane. If we allow this to grab one and pass directly through the membrane, guess what they're going to do? They're going to diffuse back through, but they're seldom going to come through the enzyme. So what's this going to do to the ATP production? It's going to greatly reduce the amount of ATP being produced, because fewer and fewer hydrogen ions are going to pass through ATP synthase. If the cell cannot form a hydrogen ion concentration, then it's going to have a rough time powering this. In other words, chemiosmosis is going to stop if these molecules don't pass or continue to pass through and are not able to establish their gradient. All right, fitness, the last section here on energetics, and I'll read through this. Explain the connection between variation in the number and types of molecules within a cell and the ability of organisms to survive and reproduce in different environments. So variation at the molecular level produces organisms with the ability to respond to a variety of environmental stimuli. This is no better example of this than with proteins, right? Proteins are wildly diverse. They are specific, so they can be tightly controlled and regulated, but they have a huge variety because of the number of amino acids and the structures in which they can be put in. Variation in the number and types of molecules within a cell gives organisms a greater ability to survive and reproduce in different environments, and that's what fitness is. Fitness is the measure at which an organism can be both surviving and reproducing, those two parts. And the measure of success is how well they can reproduce. And so with these examples here, like, if we were to put it in context for this, you've got different conditions and different responses, different protein levels being made. And the ones that can do it better will survive more and pass on their genes. And that gets us through topic 3, and I'll push out topic 4 later, which is cell communication in the cell cycle, signal transductions, etc. All right, see you a bit.